Last time we talked about regioselective enolate formation, where depending on the base you use, whether you're doing the reaction under reversible or irreversible conditions, you can make a different enolate. For example, when you use a base that is much weaker, then you would need to completely remove a proton. For example, the pKa of the purple proton and the orange proton are around 20. If we use a base that's not strong enough to completely deprotonate that proton, then you only form a very small amount of enolate, and the major enolate you form is the most stable one. So we called this last time equilibrating enolate formation. The reason it's equilibrating, oh, by the way, NaOET is the abbreviation for sodium ethoxide, where ethoxide is the conjugate base of ethanol. So what's going on here is ethoxide is removing the proton. You can tell that this isn't enough to completely deprotonate that purple proton because the pKa of ethanol is 16, whereas the pKa of the orange and purple protons is around 20. So you only form a very, very small amount of enolate, and of that enolate that you form, most of it is the most stable enolate. Your text calls this the thermodynamic enolate. I think that term is unnecessarily complicated. We just mean that it's the most stable enolate. And when you're under equilibrating conditions, the major enolate that you get is the most stable one. Okay? So that's something we mentioned last time. Alternatively, we presented a situation where you might use a base that was much stronger, that could completely deprotonate the alpha proton. We called this base LDA. That stands for a nitrogen with 2 lone pairs and a negative charge with 2 isopropyl groups on it, and the L stands for the lithium counter ion. The important thing here is that the conjugate acid of that base has a pKa of 40, so that is more than sufficient to completely deprotonate the alpha proton, and these are irreversible conditions. Now, when you remove protons under irreversible conditions, the thing that matters most is which is fastest to remove. So how can you decide which is fastest to remove? Well, if the pKa's are similar, then sterics are the major factor. In this case, the 2 different kinds of alpha protons we have both have similar pKa's. So then the only difference between them, the major difference, is sterics, and the less sterically hindered proton is easier to remove. So if you use LDA, you can hope to get this enolate. That enolate is in your text called the kinetic enolate, which simply means it is fastest to form. And when the reaction conditions are irreversible, the fastest to form is the major enolate, because once you form it, you can't go back. All right, are there questions about the difference between equilibrating enolate formation versus irreversible enolate formation? Yeah?
Is the only difference the strength of the base used?
Yeah, that's really the strength of the base relative to the acidic proton. Let me show you one place where you can get tripped up, and I've seen a couple of you get tripped up on a problem. You weren't quite sure what to do. Remember that I said steric issues are the major factor when the 2 different kinds of alpha protons have similar pKa's. Some of you have done this problem from your text, where you have a molecule like this. You've got 2 different kinds of alpha protons, and you were supposed to predict what was the major enolate. And the base was LDA. And this was deliberately tricky. The text author lured you into this mistake. The correct answer is this one. Some of you, though, applying what you had learned above, wondered, why do we not form the less substituted enolate? Isn't the one that you just formed more sterically hindered, and isn't that a problem? And the answer, the solution to that question is that sterics are only the major factor if pKa's are similar. The pKa being alpha to only one carbonyl is 20. The pKa being alpha to 2 is around, for ketones, around 9. So that's a 10 to the 11-fold, 100 billion times more acidic. That's more than enough to overcome the steric preference for the less sterically hindered enolate. So just be careful with that and understand if you're choosing between an alpha proton with, even with LDA, if you're choosing between a proton that's alpha to 2 carbonyls versus just 1, go with the more acidic proton. We're only worrying about sterics when they're similar. Yeah?
What would you consider a big difference? Would it be like between 19 and 20? Because that's thousands.
Very good. Yeah. So somebody asked that at 9 o'clock too, and the answer is certainly 11 pKa units is big enough to worry about. Probably closer to within 1 or 2. Probably. In the end, we're never going to get down to that level where you're looking at numbers and trying to decide. So, yeah. Okay. Other questions? Yes.
So in this situation, would both enolate products be the same?
Oh yes, yeah. In this situation, whether or not you used LDA or sodium ethoxide would be the same. And in fact, in this situation, even sodium ethoxide would be irreversible, right? A pKa for its conjugate acid of 16 would just completely remove this proton. Okay, good. So now let's talk about a subsequent reaction. I talked about, the title above says enolate alkylation. What do we mean by enolate alkylation? We're going to use the enolate as a nucleophile in an SN2 reaction with an alkyl halide as an electrophile. So this part won't be new because you've seen SN2 reactions before tons of times with alkyl halides as electrophiles. The only difference is you're using the enolate as a nucleophile. But notice what a difference that makes. If you use the thermodynamic enolate, it's going to react here from this more substituted carbon. If you use the kinetic enolate, it's going to react here from this less substituted carbon. So let's just imagine we bring in an alkyl halide, and let's keep it simple. Let's make it a 2-carbon alkyl halide. You can expect the enolate nucleophile, the negatively charged alpha carbon, to attack at where the bromide is, where the leaving group is. If you want to know what's going on with the orbitals, the enolate is dumping electron density from that alpha carbon into the carbon bromide sigma star. You're breaking the carbon bromide bond and you're making a new carbon-carbon bond. The product that you get looks like this. Here's the methyl group that was there. Here is the new bond that we made, and here is the 2-carbon electrophile, which I guess I'll highlight in green so that you can see where it got installed. Okay? That's called alkylation of an enolate, and it's alkylated on the alpha carbon. So we'll refer back to that in a little bit. In contrast, you can imagine doing the same reaction with the same alkyl halide, except with the the kinetic or faster to form enolate. Electrons on the orange alpha carbon would attack the antibonding orbital, breaking the carbon-bromide bond, and you would get this product where the new bond is not on the same carbon as the methyl group, but on the other carbon. In other words, your electrophile ends up here. Those two products are regio or constitutional isomers of each other, regioisomers. So we say that these reactions are regioselective because they give you preferentially the product here from the more substituted enolate or the product here from the less substituted enolate. Okay, questions? Yeah.
Sorry, so if LDA reacts with a certain molecule, it's going to always react with the less substituted area.
If the pKa's are similar, then yes, it's going to react on the less substituted side. Yep.
It seems like the tricky part is the preparation of the enolate.
Yeah. Yeah, the tricky part is the preparation of the enolate. You got to get the conditions right. Okay, what else? Yeah.
In the text, it was talking about protic versus aprotic solvents.
Uh-huh.
I'm not worried about that.
I'll mention just a little bit about it. It's not really going to be a thing where, everything was right, but you chose the wrong solvent, so now the question's wrong. But as a matter of, as a practical matter, you can totally use protic solvent here, because when you make the enolate, you're making ethanol, which has a proton, and it's reversible anyway. So protic solvents are just fine here. They actually help with the equilibration. A protic solvent would be a disaster here, because that would cause this to go back to starting material. So over here, you use solvents that are polar but don't have acidic protons. A classic example of a polar but non-protic solvent is this one called THF. You will see that abbreviation. That's just the polar aprotic solvent. Often also with irreversible enolate formation, you will use low temperature. You'll see that annotation in your text. And that's just to help make it completely irreversible. Okay, others? So I want to go back to the equilibrating enolate formation conditions. Does anybody see any problems, anything you might be worried about here? Yeah?
If there's excess base, it could react again and add another.
Okay, excess base reacting again. You're worried about maybe making another enolate on this side?
On the other problem.
Over here?
Yeah.
Okay.
An extra H from right there and add another.
So probably you need to be careful with the amount of base. What about this ethoxide? Under equilibrating conditions, is ethoxide still around by the time you introduce the alkyl halide?
Yeah.
Yeah, because at any given time you only have less than 0.01% enolate. So anything you see that ethoxide could do besides removing the proton?
That's the material.
Yeah, how about making an ether with the alkyl halide? So this is a concern that we are worried about. Now, I'm going to illustrate the worries, and then I'm going to tell you that your text, for the most part, tells you you can use this reaction as though those worries don't exist. But those worries are context for something we're going to introduce next, which is a workaround that avoids these problems. So yeah, you were worried about your ethoxide base acting as a nucleophile in an SN2 reaction. You're worried about this competing SN2 reaction that just makes this ether, which is lovely unless you weren't trying to make an ether. And then you don't have base, you don't make any enolate. The other thing you are potentially worried about is a competing E2 reaction where your base can deprotonate the alkyl halide to create an alkene. And again, this would prevent your base from doing what it's supposed to do. So these are some concerns when you try, especially when you try to do equilibrating enolate formation. These are not concerns for irreversible enolate formation, because notice how this reaction goes. LDA makes 100% yield of the enolate. By the time you get here, there is no LDA left, and so you're not worried about LDA reacting with your alkyl halide. Okay? Go ahead.
Would we also be worried about the electrons making a double bond and then kicking electrons onto the oxygen and then the oxygen acting as a group bond?
Sure. So one other potential worry that's true about both of these things, and these are potential byproducts. You would hope that they don't form, but you do have the potential for, say, this resonance structure of the enolate getting alkylated on the oxygen. In general, that sometimes happens, and if it happens, you should be prepared to know where it comes from. In general, that's an undesired byproduct. And when it happens, it's called O-alkylated, because it was alkylated on the oxygen of the enolate instead of the alpha carbon. So that is a concern. That does sometimes show up. If you saw it, you would know where it came from. in general, you try to choose your system so that's not a major issue, but it can show up. Okay, others? Yeah,
Are you missing a carbon on the worried about SN2 yet?
probably. Yep, thanks. I was running on autopilot there. I was remembering what I did at 9 o'clock where I used CH3BR instead of ethylBR for our whatever. So sorry about that. Good catch. All right. So there's a workaround for some of these issues. Again, you are free to use these reactions as though the worries were not even a thing. But you need to sort of understand them, or else it won't make any sense at all why the text is going to show you what we do next. So to set this up, suppose my desired product was this molecule. And one thing I might notice about this molecule is on the purple alpha carbon, I have 2 alkyl groups. In fact, I might call this an alpha-alpha dialkylated product. And if I compare this, my target, to what I've done before, hey, look over here. This is also an alpha-alpha dialkylated product. In other words, enolate alkylation is one way I can start with a simpler ketone and install a couple of alkyl groups. One strategy for doing this, getting this, might have been to start with the simple ketone acetone, use equilibrating enolate formation to introduce a methyl group as my electrophile. This would be the thing that gets attached. After the first reaction, I would have made this molecule. And then I could do a second round of alkylation. In this case, now with the molecule that I just barely made, I have 2 alpha protons, 2 sets of alpha protons. One side is more substituted than the other side. So I have reasonable hope of regioselective enolate formation. Using equilibrating enolate conditions, I can selectively remove the purple alpha proton to get the more substituted enolate, and then alkylate with the second CH3BR group to generate my desired product. Okay, and if you were doing this in a synthesis free response and that was your answer, that would have been great. I can't argue with it because your text tells you you can basically do it, right? But there is a better way that avoids all of these potential issues. And to remember, the major potential issue would be along the way, the ethoxide could react with the alkyl halide to make an ether. And these are things that happen all the time in the lab. Go upstairs and talk to some of the grad students on the 4th floor about reactions that looked good on paper but failed in the lab, and you'll understand what doing chemistry is actually like. So here is a workaround. And one of the reasons we're introducing this workaround is because it's going to show you an important concept that's useful in biochemistry, and that's called decarboxylation, which sounds horrible, and it will be the first time you see it. So just, prepare yourself, and we'll first do, the first part that we're going to do is pretty straightforward, and then we'll get to something that's quite challenging. So what I want you to keep in mind is our goal is this product here, all right? And the workaround is talked about in your text. It's called the acetoacetic ester synthesis. I wish they wouldn't call it that. This is acetoacetic ester. That is its name. And we're not actually making it. We're actually using it as a starting material. But I guess the focus group that looked at the textbook thought that acetoacetic ester synthesis had a better ring to it than the starting material that begins with the acetoacetic ester. At least that made for a more convenient heading in the chapter. So there you go. But this is the synthesis that starts with acetoacetic ester. Here's the leap, which we're going to come back to and see in the end, but you need to know it from the beginning. Those 3 carbons in our desired product are these 3 carbons in our starting material. The ester group's going to go away. It's going to look magic, but actually it's a pretty sensible reaction when we get there. Okay, the big advantage of acetoacetic ester is those alpha protons in the middle, which I guess I should have made purple, are very acidic. Their pKa is, I forget, but it's somewhere near 13, I think is the right number. In contrast, the protons that are on the other alpha carbon have a pKa of 20. So you don't have to worry about regioselective enolate formation at all. You don't have to worry about steric hindrance. The pure acidity of those protons that are alpha to 2 carbonyls make them the most acidic protons. The other big advantage is I don't need to use a very strong base at all. In fact, I can use sodium ethoxide. And I can use this and it will give me in 100% yield the enolate of acetoacetic ester in which I have a negative charge on the purple alpha carbon. And that is because the conjugate acid of my base, ethoxide, is ethanol, which has a pKa of 16. So you can do the math. We're favoring this by a factor of more than 1,000, or about 1,000 anyway. Okay, one quick thing. I deliberately chose sodium ethoxide as my base to match this group. What do you think could potentially happen? What's a potential undesired side reaction if I used OH minus? Go ahead. Accidentally attack
the tail end.
Okay, so removing this proton is a possibility, though the fact that these protons are more acidic means we don't worry about that. These are more acidic by about 10 million times. There's something else. Go ahead.
Do you attack that ketone? Well, not ketone, but the ester there.
The ester. Sure, right. If you attack the ester with, say, hydroxide, do you see the problem? Is now I've got 2 equally good leaving groups relative to a negatively charged oxygen. I might hydrolyze the ester. So to prevent that, I just use ethoxide as my nucleophile. So if I do attack the ester carbon, I get a tetrahedral intermediate like that, and it doesn't matter which group gets kicked off. I end up with the same thing. So that's a cute little strategy. It's not necessarily that important. I just wanted to point it out. And it's also not like there's going to be a question on a test where there's some big thing here, and if you don't match it exactly, you get the problem wrong. Just understand this is why we're using that as opposed to something else. Okay, so you make the enolate, then, and notice at the once you make that enolate, there is 0% ethoxide left. So now we are totally sidestepping the worry about the competing SN2 reaction with the ethoxide. Now the enolate reacts with the alkyl halide, and we've got one of our 2 methyl groups installed on the alpha carbon, sorry, my cruddy penmanship, installed on the alpha carbon of my starting material. Okay, so looking ahead to where we are, we've got one of our 2 methyl groups. At that stage, I can simply do it again. 1 molecule of sodium ethoxide and another molecule to make the enolate in 100% yield, and only then do I introduce the alkyl halide, and that will install the second alkyl halide on the alpha carbon to give me this molecule. So far, aside from the fact that the molecule is a little bit bigger than we saw before, we're just doing enolate alkylation, and we're taking advantage of the fact that the purple alpha proton, or the protons on the purple alpha carbon, are especially acidic. That's allowing us to completely form the enolate in 100% yield so that the alkyl halide never has to see ethoxide. Okay, with me so far? Anything you want to ask? So where we want to get to is this product. Notice the differences between this product and this one. Both are now alpha-alpha dialkylated, But the one on the right and the bottom has this ester group, which is not present in the starting material. So we have to get rid of the ester. This is going to look like literal magic, like somebody reaches in and pops the bond off like you would with a model set, and suddenly everything's okay. It's not, and there's a mechanism for everything that's going to go on here. The conditions to get from here to here are actually very simple. But there's three different things that go on under these conditions. I need to move this up somewhere. The conditions are aqueous acid, a strong aqueous acid, and heat, which looks like a fire triangle, which is why we use it as the symbol of heat, I guess. Okay? So there are three things that happen, and we'll take you through them one by one. The first is what you expect to happen when you put an ester in an aqueous acid, and that is acid catalyzed ester hydrolysis. I'm not going to show you the mechanism of that today because that is a mechanism that you learned in chapter 16. If you need to see it, go ahead. If you need to review it, go ahead and take some time over the next couple of days to look at that mechanism and make sure you understand it. Basically, that the acid would protonate the carbonyl of the ester, water would attack the oxonium ion, you would do proton transfer to change who's the good leaving group, and eventually ethanol would leave. In the end, you go from what used to be your ester to the expected hydrolysis product, which is this acid. And structural patterns here, the ketone on the beta carbon makes this a beta keto ester. Similarly, we would call this one a beta keto acid. And beta keto acids are an important structural pattern to recognize in chemistry and biochemistry because they have some interesting properties that are really useful. And one of those is that beta keto acids spontaneously decarboxylate. In fact, some of the CO2 you are exhaling right now comes from spontaneous decarboxylation of a beta keto acid in the citric acid cycle that's going on inside your mitochondria. So this is going on all the time. We're going to see it again and again, and this is a convenient place to show you, to introduce the concept to you. So the second step is called spontaneous decarboxylation. Going to be a lot of arrows going on in this process, and I'll try to walk you through them. I'm going to redraw the beta keto acid in a slightly different way. I'm going to angle the carbonyl down here of the acid and put the OH group here. If I do that, you can see that the proton of the carboxylic acid group is actually close to the carbonyl of the beta keto group, right? And in fact, you might expect that carbonyl oxygen to even be hydrogen bonding with that proton from the acid. And it's not too much of a stretch to imagine, well, what if the carbonyl oxygen on the beta carbon takes that proton entirely? What then happens is if you stopped, you'd have a negatively charged oxygen here, the conjugate base of the carboxylic acid. But instead of forming that negative charge, these electrons are going to kick down and form a new pi bond between carbon and oxygen. That would actually form CO2. In order to do that, though, we have to break the bond between blue carbonyl carbon and blue alpha carbon. Those electrons will come over to here to form a pi bond between alpha and beta carbons, and then we'll break the carbon-oxygen pi bond. In this process, the atoms I'm highlighting in blue become CO2, and the atoms that I'm going to highlight, I guess, in green, are basically a leaving group. In other words, you've never seen a carboxylic acid do this, but essentially we're seeing a carbonyl kicker-offer type reaction. The oxygen on CO2, the oxygen on the carboxylic acid does the kicking off. And what gets kicked off and what's left behind on the other side is this enol, which is a neutral leaving group. So that's why this can happen, is because CO2 leaves and what's left behind is neutral. Okay, this is what happens when you do decarboxylation under acidic conditions. You can also do decarboxylation of beta keto acids under basic conditions. I'll show you that too, and that'll give you another sense for what's going on here, but let's just pause. Any questions you have there? Yeah?
Will that enol tautomerize?
Yeah, so that's the third step. That enol has a keto form that's more stable, and it's going to tautomerize. Yeah?
Why is the oxygen able to strip off the hydrogen from the other oxygen? Why is that bond more likely to happen?
Why is that able to happen? The thing that probably pays for this, first, that oxygen's hydrogen bonding anyway. The thing that pays for it is that CO2 leaves and is a gas and bubbles off, and that dramatically increases entropy. So that's why this is so downhill in free energy. What else? Okay. Well, let me show you that decarboxylation can also occur under basic conditions. So beta keto acids also decarboxylate under basic conditions. This is relevant because most of the decarboxylation reactions that you see will happen not in biology, will happen not for the acid form of the carboxylic acid, but its conjugate base. So here is the carboxylate of a beta keto acid. What can happen here is that the negatively charged oxygen kicks down to make the new carbon-oxygen pi bond, and then the bond between the carbonyl carbon and the alpha carbon breaks, and electrons move on to the alpha carbon as a negative charge. You'll get CO2, again, bubbling off, and then what's left behind is not an enol now, but an enolate. But that is okay because the enolate is stabilized by resonance and can quickly pick up a proton in biology from some source. So we will see both types of, mostly in biochemistry, you will see this kind of decarboxylation of a beta keto carboxylate. For today, with the acetoacetic ester synthesis, this can happen under acidic conditions too. Okay? So notice what that does. Remember, our goal was to get to this product. We got rid of the ester, but we're still not at our product. We've got all of the pieces in place, but there's a difference, and you've noticed, some of you've noticed this already. What we've got here is not our product, but it is the tautomer of our product. It's the enol form of our product. So the final step that happens is tautomerization of the enol to the more stable keto form. So here is the enol, and it's going to form spontaneously the more stable keto form. And this is my desired product. Again, the 3 carbons that came from acetoacetic ester, the 2 alkyl groups that we installed. Tautomerization you can draw a mechanism for tautomerization under both acidic or basic conditions. Some of you have done this before. The easiest way to do this under acidic conditions for this enol is to have these electrons kick down and form a pi bond, and the pi bond between carbonyl carbon and alpha carbon to break and get protonated on the alpha carbon. That would take you to this oxonium ion. And then to generate the keto form, you would simply regenerate your acid catalyst by deprotonating the oxonium ion, and there you go. So that's one way to tautomerize an enol to the more stable keto form. All right, so that was a lot, right? The alkylation first of the acetoacetic ester, then we did a second round of alkylation to install the second alkyl group. Then we used simple conditions, water, heat, and acid, to do three things: ester hydrolysis, spontaneous decarboxylation, and then tautomerization. Okay, questions? All right, let's do this. Oh, go ahead.
What was the whole thing called?
The whole thing, the whole shebang from the very beginning was called the acetoacetic ester synthesis. Okay, let's do another example from the forward direction. Then we're going to learn to think backwards, because as you've learned on synthesis problems, thinking backwards is an important part of the problem. For this example, we're going to use a slightly different starting material. This one is, instead of being a beta keto ester, this is called a beta diester. This specific example is called malonic ester, and your text will call what I'm about to do the malonic ester synthesis. You should not think of it as an entirely separate thing from the acetoacetic ester synthesis. It proceeds in exactly the same way, though the product is different in a way that I will show you that's completely rational and completely predictable. So for this molecule, I'll take you through a forward synthesis. We're going to generate an alpha-alpha alkylated molecule, and then I'll show you what happens after ester hydrolysis, decarboxylation, and tautomerization. So let's imagine that, again, we're going to use sodium ethoxide to deprotonate the purple alpha carbon, and then we're going to add an alkyl halide. And for fun, let's do 3 carbons, right? We're living on the edge with 3 carbons. So that's going to install, it's going to make a new bond in our beta diester. Here is the new bond in red, and then we need 1, 2, 3 carbon chain there. There's still a proton there. That is our first alpha group, first alkyl group on the alpha carbon. Then let's imagine, maybe I'll slide things over to give ourselves a little bit more room. Then let's imagine a second reaction where I deprotonate the alpha carbon, and this time I'm going to use something that is a little bit more horrible. Let's use a benzene ring with a bromo group. All right, and so that should deprotonate the purple alpha carbon, generating an enolate, which I'm not drawing. And then that enolate should react with the carbon that has the leaving group on the alkyl halide, and that should make this new bond. And then the benzene ring would be here. So in green, we'll have the 3 carbons of the first alkyl halide, and then let's do pink for benzene. Here's the benzene ring, and its sort of attached carbon. Okay? That's so far pretty similar to everything we've done previously. Now let's take that molecule and let's do the strongly acidic aqueous conditions with heat, and that's going to do ester hydrolysis, decarboxylation, and tautomerization. So let's draw each of the intermediates along the way. Let's draw the products of ester hydrolysis. The difference here is I have 2 esters. In the acetoacetic ester, this one was a methyl group. Now I've got two esters. Both of them are going to get hydrolyzed to carboxylic acids. So the first thing that happens under those strongly acidic aqueous conditions is that I hydrolyze both esters to the carboxylic acid. So now instead of a beta diester, I have a beta diacid. The second thing that's going to happen is decarboxylation. I'll actually draw this mechanism for you so that you can see what's going on. We'll draw it exactly the same way as we drew the decarboxylation mechanism previously. That is, the carbonyl oxygen on one of the carbonyls is going to steal the proton from the OH group on the other acid. Electrons will kick down to form a new carbon-oxygen pi bond. That forms CO2. Then the rest of what happens is we form a new carbon-carbon pi bond between alpha and beta carbons, and then this carbon-oxygen pi bond breaks. The two products are CO2 and then an enol. And that enol will look almost exactly like the enol that we used before. Only the other thing here is an OH group instead of a methyl group. If you go back in your notes and compare, for the acetoacetic ester synthesis, this used to be a methyl group, but now it's an OH group. That doesn't change anything. The decarboxylation happens in exactly the same way. And then finally, the last step is tautomerization. And I won't draw the mechanism for this. I'll just draw for you the final product. Which looks like this. Okay? So this is our final product. Notice that from our starting material up top, which was malonic ester, these groups, the carbonyl oxygen and the alpha carbon and the carbonyl carbon, are these three atoms in the product. The green atoms in the product come from one of the alkyl halides. The pink atoms in the product come from the other alkyl halide. The OH group here comes from the water that we use for the hydrolysis reaction. Okay. So now let's talk about thinking backwards. When you think backwards, you first need to look at a desired product and recognize some structural features about it that connect it in your mind to the reaction that you think maybe made that product. I've highlighted that a couple of times today, and will do so again. Notice that this product has two alkyl groups on the alpha carbon. This is an alpha-alpha alkylated carboxylic acid, two alkyl groups on the alpha carbon. That's a useful structural pattern, and if you see it, you can be thinking, I could have made that starting with the beta diester or the malonic ester. Similarly, if you see here our product, which was an alpha-alpha dialkylated ketone, and in this case it's a methyl ketone, that is a signal that you could have made that from the acetoacetic ester. So let's do a challenging example, and it's going to be so much worse than these other ones because we're going to make a ring that wasn't there in the starting material. So we're actually going to do this thinking backwards. Okay. So you could have made that from, this is your target, this is where you're trying to get, and you need to make this from this starting material and any other reagents. So the first thing to do is to notice that this is an alpha-alpha dialkylated methyl ketone. Where's the methyl group? Right here. Where's the other alpha carbon? Right here. And does it have two alkyl groups on it? Yeah, one is here, one is there. So that's the structural pattern. If you recognize it, then you've already said that these groups highlighted in gray, those 4 atoms, are these 4 atoms in the starting material. In purple is the alpha carbon I need to alkylate. And then maybe I'll just go ahead and highlight in blue is the stuff that's going to come off as CO2. So if you recognize that pattern, you already have a synthetic plan in mind. Now the question is, what is the alkyl halide that I should use? I'm guessing that that's kind of challenging as it is looking at that 5-membered ring. So we're going to do a magic trick. And that is we're going to erase one of the bonds. Now, is it easier? Okay. What alkyl halide would I use to do that? Presumably, I would do that one once, right? And then a second time I would deprotonate, and then I would attach that one, right? That would get me where I need to be. So what if they're both attached together? Doesn't that work? It should. Ta-da! Right? Notice 1, 2, 3, 4 atoms. The alpha carbon is bonded to atom 1 and atom 4. So the alkyl halide I need has a leaving group on 1 and another leaving group on 4. Okay, so it's as easy as that. What you're going to do is use sodium ethoxide to make the enolate. Then you're going to use this 1, 2, 3, 4 carbon dialkyl halide, dihaloalkane or whatever. And you're going to make one of the 2 bonds here in red. Then this is carbon 1, 2, 3, 4 with the Br here. Then you will use the second equivalent of sodium ethoxide. That will deprotonate the alpha proton that's especially acidic. And then you can hope for an intramolecular substitution reaction in which that purple alpha carbon attacks the antibonding orbital there and makes the bromo leave. That closes the ring. And if you count the atoms correctly, you should get to the right size ring. Then getting to that product is just a matter of ester hydrolysis, decarboxylation, and tautomerization. We went through that whirlwind wicked quick, and the magic trick looks still miraculous to you, so you need to go through this more slowly until you convince yourself that it works, and then push it one step further, see if you can make a 6-membered ring. Yeah?
On the 4-carbon ring, if you had a bromine on carbon 1 and a bromine on carbon 2, would it make a new pi bond?
If you'd had a bromine on 2 and a bromine on 3, would it make a new pi bond? I don't know. Come up afterwards and we'll talk. Thank you all. See you Friday.